Hello, I'm Matthew Jarron, Museum Curator at the University of Dundee, and welcome to the latest in a series of short films in which I talk about some of the highlights of the University Museum collections. Now, Dundee's always been renowned for its interdisciplinary research, and today I want to talk about some really important work which began here in the 1940s when we were University College Dundee, which brings together physics, chemistry and biology, and which played a crucial part in unravelling the mysteries of DNA. And that is Dundee's often forgotten role as a key centre for X-ray crystallography. So crystals, as I'm sure you all know, are solid materials whose constituent parts, such as atoms or molecules, are precisely arranged in an ordered structure, forming a crystalline lattice. Examples of some of the larger crystalline forms include diamonds, snowflakes and salt crystals, and these are crystals that were actually grown in the chemistry department here in the 1930s. Now, there had been serious scientific study of crystals since the 17th century, but it was only when X-rays were discovered in 1895 that the possibility arose of determining the details of their internal structure. In 1912, a German physicist called Max von Laue discovered that if you shone a beam of X-rays onto a crystal, the X-rays would diffract into many directions, each of which had very specific and measurable angles and intensities. Shortly after, an Australian-born physicist working in Cambridge called Lawrence Bragg worked out why this happened, and with his father William Bragg came up with an equation to explain it. It thus became possible to use these measurements to produce a three-dimensional picture of the density of the electrons within a crystal, and from that work out its atomic structure. The implications of this were immediately realised. Discovering the structure of even the simplest inorganic crystals and minerals revealed fundamental laws of physics and chemistry that had not previously been confirmed. A whole new field emerged, known as X-ray crystallography, which still to this day is the primary method for characterising the atomic structure of new materials. In Dundee, the story really starts with this chap, George Dawson Preston, who was Professor of Physics from 1943 to 1966. Preston had worked at the National Physical Laboratory at Teddington, where he specialised in X-ray diffraction work. As part of this research, he actually helped to build the UK's very first electron microscope, and he then purchased a second from Metropolitan Vickers, which he brought to Dundee to continue his research. We no longer have that, but this is a similar one that's in the Science and Industry Museum in Manchester. What we do still have in our collections are some of Preston's original photographs and slides of X-ray diffraction patterns. Preston was keen to apply these techniques to crystalline structures in biology. And so here are some of his glass slides with samples of Tradescantia or spiderwort and allium root tip. Preston was keen to bring other X-ray crystallographers to Dundee, and in 1948 he made two significant appointments. One was a young junior lecturer called Gavin MacDonald, this is him later in life, who completed a PhD on crystallographic investigation of organic compounds and would go on to publish the structure of several different crystal structures. He moved to the West Indies in 1957 to the University College in Jamaica, where he established an X-ray crystallography unit in order to continue his research. He returned to Dundee in 1962, and his many publications include a key textbook on physics for biology and pre-medical students. In 1974, he became the university's first vice-principal. The other key appointment that Preston made in 1948 was this chap, John Eyeball, who had begun his interest in X-ray crystallography at the Royal Institution in London, studying under William Bragg, who I mentioned earlier. Eyeball was initially appointed to the physics department, at that time based in the Carnegie Laboratory, and here he is with some of the other physics staff and research students, sitting next to Preston in the middle there. But he later on transferred to the chemistry department in the Carnelli building, where he spent the rest of his career. In 1950, Eyeball received a Royal Society grant to purchase this camera for the university, which we still have in the collection today. This is called a Leeds Weissenberg camera, and it was a state-of-the-art piece of kit for X-ray crystallography work. It works by rotating the crystal during the photography, while also synchronising the movement of the film, and it does this again and again until you can get a suitable intensity of exposure. The results allow for far quicker and easier interpretation of X-ray diffraction patterns, and this camera was of such high quality that it actually remained in use here for almost 50 years. Eyeball used it to create diagrams like these, taken from the photographs, which he then translated into electron density maps like these, many of which we have in the collection. In 1952, Eyeball was made a research fellow in the British Empire Cancer Campaign, and much of his research focused on X-ray studies of carcinogenic compounds. He made major advances in cancer research and actually gave his name to the Eyeball Index, which measures the relative potency of carcinogenic compounds. One of the main ones he studied was benzopyrene, which is found in coal tar, car exhaust fumes and cigarette smoke. This is Eyeball's original electron density map of benzopyrene, which was then published in Nature in 1956. Eyeball published numerous research papers on X-ray crystallography, many in collaboration with other colleagues at the university, mostly in the physics department, such as Douglas Young and Pat Tollin. One hugely important area of research for X-ray crystallography was DNA, and in 1957 a highly significant new arrival came to Dundee who played a crucial part in the DNA story, Herbert Wilson. So I'm sure you'll all be familiar with the iconic double helix structure of DNA, and that was worked out by studying X-ray diffraction patterns. The discovery was announced in a very famous paper by James Watson and Francis Crick of the Cavendish Laboratory, which was published in Nature in 1953 and ultimately won them the Nobel Prize. This is the diagram that was in that paper, and we have this wonderfully low-tech model of it in the collection that's held together with paper clips. But that paper was accompanied by two others in the same issue, one by Rosalind Franklin and Raymond Gosling, and another by Maurice Wilkins, Alex Stokes and Herbert Wilson. So Wilson was one of a team of researchers based at King's College London and working under Wilkins, who used X-ray diffraction to produce the key evidence that Watson and Crick relied upon. Interestingly, Gosling also came to Dundee to do X-ray crystallography, but was soon lured away to join Gavin MacDonald in the West Indies. Wilson, however, would remain here for the next 26 years. He worked with the rest of the Dundee team to determine the structures of many of the component parts of DNA, some of which I'm not even going to attempt to pronounce. One of the ones I can say, though, was guanine hydrochloride dihydrate, which was published by Nature in 1963, and we have this model of the molecular structure of it, which Wilson and Eyeball made. Another molecular model that we have is this one of sperm whale myoglobin, which was made for Pat Tollin around 1966. Now, Tollin had developed a pioneering system whereby a model could be matched to reality by rotating and repositioning, and thus determining the position of molecules within the crystal cell space. Sperm whale myoglobin was chosen to test the system, and Tollin worked with a research student called Jenny Gorski to determine the position and orientation of the myoglobin molecule within the crystal. And the same method was successfully applied to seal myoglobin as well. The X-ray crystallography work continued with great success into the subsequent decades, with new colleagues joining, such as John Lowe. Here's another important piece of kit. This is a goniometer, which the university bought from West Germany, used to measure the angles between crystal faces. It's worth noting also that Herbert Wilson continued to collaborate with his DNA mentor Maurice Wilkins. They published this paper together in 1969, which, as you can see, was originally communicated by Francis Crick. In 1966, Wilson also wrote one of the key textbooks on diffraction of X-rays by proteins, nucleic acids and viruses. And it was this last area that Wilson and Pat Tollin shifted their research into in the 1970s, using X-ray diffraction to study the structure of viruses. And in particular, they collaborated with the Scottish Crop Research Institute in Invergowrie, now the James Hutton Institute, to look at plant viruses, including potato virus, tobacco rattle virus, Narcissus mosaic virus, and various others. In the 1980s, Tollin also turned his attention to the devastating AIDS epidemic, using X-ray crystallography to determine the three-dimensional molecular structure of the zidovudine drug, which was the first approved treatment for HIV. For nearly half a century, Dundee was a world leader in X-ray crystallography research, and the collections that we have are physical reminders of groundbreaking work that contributed to our modern understanding of DNA, cancer, viruses, and many other areas. Well, that's all I have time for just now. Hope you found that interesting, and join me for another film coming soon. Bye for now.